I'll give you a very brief story on choline, which was discovered in the mid-1800s. The role of choline on intermediary metabolism, its effects on fatty liver, which is really the main reason why choline was initially used in human diets, but it has many other roles. And then I'll tell you effects during the transition. I will touch on the concept that not only overconditioned cows respond to choline, and I'm going to skip this part here just because of the sake of time. And hopefully I'll make the case that choline is a required nutrient as it is for other species, as it is in baby calf diets. So as I mentioned to you, choline was really discovered in France and in Germany by pharmacists and chemists. But it wasn't until early 1900 that it became really important. And it was really something that they were not expecting. During the process of discovery of insulin in Canada by Frederick Banting, they used this animal model in which they pancreectomized dogs. So they would remove the pancreas to study insulin, eventually to study insulin, as they eventually named. And one of his graduate students, Charles Best, noticed that those dogs developed fatty liver. And in the early 1930s, they found out that if they fed pancreas to dogs, or eventually when they fed lecithin, phosphatidylcholine, they were able to reverse the fatty liver case. So that's where the story of choline really started as an important nutrient in mammalian diets. But in the late 70s, early 80s, work out of England by Derek Lindsay and colleagues showed that in the case of ruminants, almost all the choline is synthesized de novo, okay, through de novo synthesis. And it was speculated that the reason was because the microbes were degrading choline to these trimethylamine oxides in the rumen, which was eventually confirmed by work from Rich Erdman at the University of Maryland, in which he showed that there's almost no dietary choline escaping microbial degradation. You know, less than 5% of that escapes microbial degradation. So this makes the case that if we want to study choline, either we do infusions or we developed technology to protect from microbial degradation, which creates some complications, as you can imagine. So how is choline used? There are multiple pathways for use of choline, and this is just a simplified version. Choline is this trimethylamine compound that has nitrogen and methyl groups, and it can be acetylated in the central nervous system or in peripheral nerves to acetylcholine, which is a neurotransmitter that's important for propagating nerve impulses, or in the neuromuscular junction to propagate a nerve impulse to a muscle for contraction. Choline, when absorbed from the diet, it can be phosphorylated in multiple tissues to synthesize phosphatidylcholine, which is this phospholipid here. There are many of them according to the composition of fatty acids they have. And obviously, this is very important as a component of every cell membranes and organelles. Or it can be oxidized to produce betaine. And betaine is capable of donating methyl groups in the one-carbon metabolism. Okay? So it can donate methyl groups to homocysteine through the action of this enzyme here, betaine homocysteine methyltransferase. And this can be used to resynthesize methionine through the action of this enzyme, methionine synthase. And methionine then condenses with adenosine to form S-adenosylmethionine, which is an important methyl donor to methylate genes and to silence genes in most cases. So it creates this cycle here. And phosphatidylcholine can be synthesized through this transmethylation reaction through this salvage pathway that usually takes place in the liver. So if animal consumes and choline escapes degradation, you can synthesize phospholipids through this pathway. But if we feed unprotected choline, most of it is degraded, so very little is synthesized through this pathway, and most of it is liver dependent through the salvage pathway. Okay. So obviously, we think that choline is a required nutrient, and we can make the case it's pretty important. I'm not sure if we can say it's essential because it can be synthesized, particularly the phospholipid can be synthesized de novo. But there are requirements established for humans, like for Scott here. He needs 0.5 gram per day. If he eats too much of that, it creates a problem because it accumulates these trimethylamine products that are linked to cardiovascular disease. But if he doesn't eat enough, then it's a problem for fatty liver. In poultry, nutrition is well established, how much is needed. For a laying hen, it's like an adult human. For a young chick, it's only 20% of that. A lactating sow requires a lot more just because of the amount of milk produced pre-weaning. But when it gets to the lactating cow, we really don't know, and there are limitations to that. And I think, in my view, the limitations is that we really don't know how much gets absorbed from different rumen protect choline products because they differ in their ability to protect and the ability to release. And we don't have those response experiments demonstrating the exact amount that optimize a particular function. Let's say if it is milk yield, optimize energy cracked milk yield. Or even if it is fatty liver, because the responses in the literature are linear, they haven't been curvilinear or quadratic that you can establish an optimum amount. So those limitations make it not be part of our required nutrients in the diet. Now, when cows get close to calving, concentrations of phospholipids in tissues and in blood, in this case is blood, decrease. And the assumption here is it's being taken up for synthesis of mammary tissue, replenishment of mammary cells that replicate in this late gestation period, perhaps uptake by the fetus, the pregnant uterus, but also because of synthesis of colostrum in milk that takes up phospholipids, and associated with the reduced dry matter intake. Remember, this is being synthesized de novo from methyl donors because of the low intake of choline. So very likely inadequate intake of or increased needs of methionine that are being used by the mammary gland would limit the supply of methionine to be methyl donor for synthesis of phospholipids. So all of those things together probably play a role in this decrease, which is pretty typical of most nutrients as the cow gets close to calving and initiates lactation. This is the same time when cows are more prone to develop hepatic lipidosis. And this is just a compilation in the literature in which hepatic tissue was collected for biopsy, and then in the biopsy was quantified triacylglycerol. A typical well-fed liver, or a typical liver of a well-fed cow contains 1 to perhaps 3% triacylglycerol. But once the cow undergoes lipomobilization, it accumulates there. And you can see that about half of the cows, if you do the math here, have between 5 to 10, and then approximately 15% of the cows will have very severe lipidosis in the first week after calving. So 50% of the cows have this increased concentration of triacylglycerol. The total lipids also increase, but triacylglycerol is the one that increases the most. And when it accumulates, it forms these fat droplets that disturbs the behavior of the hepatocyte and compromise hepatic function when in excess. So obviously, excessive lipidosis is not desirable for cows. Just to show you the role of choline in reducing hepatic lipidosis, this is an experiment in which we assign 110 prepartum Paris cows to one of 5 treatments. So cows were assigned based on their body condition score at 232 days of gestation. Okay, and the treatments were a control in which we fed just the coating material, but no choline in the diet. So just what is used to coat choline in the Rumen Protect choline product. And then they were fed 13 grams of choline ion from a product that has low concentration of choline in the whole material that was fed. Or we doubled the dose, almost 26 grams of choline ion. So this is about choline chloride is about 75% choline ion. The other 25 is chlorine. So this here will be 30-some grams of choline chloride that is in a room-protected form. And then we use the exact same amounts, but from a different product that contain a larger concentration of choline chloride in the product. Okay, so we have the, as you're going to see the data, we have the effects of choline against control, and then we have a factorial arrangement here. We have two products. This is one product, this is another product, and we have two concentrations, or two amounts, 12.9 grams versus 25.8, and then the interaction term, which is this plus this versus the average of these two. And that's how I'm going to show you the data. So these cows had 5 days of ad libitum intake, and then we restrict the amount they were fed to about 42% of the net energy for lactation to meet the needs for maintenance plus fetal growth to induce fatty liver. And we induce fatty liver in 80% of the cows with more than 5% triacylglycerol with this protocol here. Okay, so we collect liver tissue on day 6 and 13 of the experiment. This is the day 13 data, so after they underwent feed restriction. So here is liver triacylglycerol as is or on a dry matter basis. So you can see here that the cows that were in the control, their triacylglycerol content was 9.3% of the hepatic tissue. And then feeding choline, which is the combination of all these four, reduced the concentration of triacylglycerol. As we fed more, it further reduced that. So that's the effect of those. And there was no difference between the products. It didn't matter whether we expressed the data on as is or on a dry matter basis. On the other hand, and this is a quite consistent response in multiple species, and the cow is no different, the concentration of glycogen actually increased. Here you can see the effect of choline, which is the average of these four versus this one here. Then you can see that it further increased as we increase the dose of choline ion. So glycogen increase and triacylglycerol decrease with feeding choline. So if you look at the ratio of triacylglycerol to glycogen, which is being suggested as a proxy for the degree of fatty liver, it was a lot more favorable in the cows that receive Rumen Protect choline, particularly when they received the larger dose of choline compared to the control cow. So this is not the first experiment to show this. This is just another one that shows this effect when we use the dry cow model. This was initially shown by Rick Grammer in Wisconsin. Okay, so then we had a second experiment in which we fed cows either 0 or 25.8 grams of choline ion. And then after 9 days of induction of fatty liver, we thought that this would disrupt gastrointestinal integrity such that it would affect digestion. And then on day 9, we fed those cows 450 grams of fatty acids from a high palmitic acid product. And we collect blood multiple times, but then we collect fecal samples multiple times just to look at fat digestibility. These cows were eating very little, so they were consuming about 60 grams of fatty acids from the TMR, and then we dose with a large quantity of fatty acids, relatively speaking. Okay, and then we analyze fecal samples for indigestible NDF as our internal marker, and then for fat, and then we collect supramammary lymph and analyze for multiple things, and I'll just show you briefly those results. So here is the digestibility, the apparent total tract digestibility of fat. It increased with the cows that were fed 25.8 grams of choline ion in a room-protected form. So there may be some possibility here we can discuss, I guess, during the questions and answers. This is the serum triacylglycerol. Concentrations were greater for the cows that were fed rumen-protected choline. And we measured the metabolism of the lymph fluid, and there was a greater concentration of choline in the supramammary lymphatic vessels that drain the mammary gland of those cows. So we're able to manipulate concentration of analytes in blood, but perhaps important is the fact that we are able to enhance digestibility of fat here. So presumably, the effect of choline reducing fatty liver is by its action on promoting the export of triacylglycerol from the hepatocyte. Because choline is important as a component of synthesis of phospholipids, and phospholipids are pretty important in terms of packaging things that are hydrophobic to be transported into bloodstream. So they are one of the components of lipoproteins. We thought, oh, let's see if this is really the case. And to do that, we infuse intravenously this non-ionic detergent that coats the lipoprotein particles and induces hyperlipidemia in animals. And it does that by preventing lipolysis of lipoprotein particles. So this is how the experiment was done. These cows went through a fatty liver induction protocol. There were 33 cows. They received either no rumen-protected choline or 25.8 grams of choline ion in a rumen-protected form. And then we infused this Tyloxapol on a particular dose based on data from mice, rats, guinea pigs, and some initial work with cows in the 1970s. So we extrapolate that to our cows today. And we infuse them here, and then we collect blood samples sequentially. And the idea is that this would coat the VLDL particles and prevent lipolysis of the triacylglycerol that's in the core by lipoprotein lipase. And then this triacylglycerols accumulate, and you can look at the rate of accumulation as a proxy for the rate of VLDL secretion or nascent VLDL secretion by the liver. These cows were off feed for about 36 hours. So they were fed the day before only 70 grams of fatty acids. So there was very little gastrointestinal fatty acid coming that would justify as being chylomicrons in the bloodstream. Okay, so just to show you the results. So this is the rate of accumulation of triacylglycerol over time in the control cows. And this is the rate of triacylglycerol accumulation in blood from the cows that received 25.8 grams of choline ion. Just to illustrate this, maybe easier to see, we look at the area under the curve, which is this area here for the control, and we look for the other two treatments that really don't differ. And you can see that the cows that receive rumen-protected choline had the larger area under the curve, meaning that there is more accumulation of nascent triacylglycerol coming from VLDL, suggesting that the hepatic secretion of VLDL is enhanced, which is what's been shown in many other species. So the cow is no different than other mammalian species. Now, obviously, this has to benefit cows. So a few years ago, we collect data from literature of published randomized controlled experiments in which rumen-protected choline was fed starting before calving. And this is one of the results of that meta-analysis. This is a forest plot. Each line represents a comparison between some level of choline ion in a rumen-protected form against a negative control within that experiment. So you can see this is an experiment here at Purdue University. This is Cornell. This is in Italy. So there are many experiments here, all the way down to Florida at the end here. So the central line means that there's no difference between the treated and the control. Okay, so if the point estimate falls in the central line. The point estimate is this black dot that you see here. The horizontal line is the 95% confidence interval. and the square is the weight of that comparison in statistical analysis and the meta-analysis. So you can see here more precise experiments that have either more cows or smaller standard errors of the mean. They have more weight in the analysis. So important point here is to look at the external validity of the data. You know, different experiments represent different farms, different managements, different diets, okay, as well as different products in this data set. You can see that most point estimates fall to the right. The response being measured here is energy-corrected milk. So there's been an increase in energy-corrected milk. You can see here 0.4 standard deviations. This represents an average of 2.2 kilograms, about 4.8 pounds of energy-corrected milk. And it changes with the dose that was fed. Second important point is that there was heterogeneity in the data, which is this I-square statistics here, meaning that the level of response changed with the experiment that was done. Okay, I'll show you 2 points that are important here. You've seen one of them because Mike already showed you. One is the dose. As the intake of choline increased, choline ion, the response increases, whether it's milk or energy-correct milk. You can see the slope's positive. And the second one that we found was linked to the supply of metabolized methionine in the postpartum diet. So the way you read this slide here, and I'm going to show the energy-correct milk just for simplicity. So these were diets that, using the old NRC 2001, had 1.7% of the metabolized protein as methionine, metabolized methionine, so digest methionine, all the way up to almost 2.7. Okay? So if you look at the bulk of the data, it's here in the middle. So between 2 to, or 1.9 to 2.25, 2.3% methionine. Okay. The black circles represent the control choline diet, so 0 grams of choline ion. This diamond shape, okay, represent the 24 grams of choline ion supplement during the transition period. So you can clearly see that when the diets were very marginal in methionine, there was a big response in energy-correct milk if you were to go from 0 to 24 grams of choline ion. If the diet was very high in methionine, there was still a response, but it was reduced. But there's still a response here, 1.5 kilograms of energy-correct milk when you go from 0 to 24 grams of choline ion, which is no surprise because of their relationships. Okay, there were other things. We found 2 things in this data set. There was a reduction in retained placenta and a reduction in mastitis when cows received choline during the transition period. We didn't see effects on other responses, but just to make the point, in this particular meta-analysis, we had 20 papers, 21 experiments, 1,300 cows, but only half of the papers reported health data. So the robustness of the data for disease was not as strong as for production, just because people don't report those results in scientific literature sometimes. Okay, so there's a carryover effect of supplementing choline. These are 2 experiments that we've completed at the University of Florida. Choline was fed from 3 weeks, from 255 days of gestation to 21 days postpartum. And you can see that the cows continue to produce more milk after choline supplementation stopped. In this first experiment here, 2 kilograms more. In the second experiment here, another 2 kilograms more. So there is a carryover effect, which is no surprise. You do good things for cows in early lactation, they respond continuously past the period of supplementation. So we think that this has a lot more to do with other things than just fatty liver. Remember that during this transition period, cows have low phosphatidylcholine, so maybe we are able to make up for some of this drop here by supplementing choline during the transition, and Marcus Zenobis has shown that increase in phosphatidylcholine in plasma of cows that were fed choline. But choline is also substrate for this enzyme here, choline kinase, which phosphorylates choline in the pathway in multiple tissues, including the mammary gland. And choline kinase is a marker for mitotic activity in memory cells. Actually, it's a marker for diagnosis of memory cancer in humans. So it's possible that by providing substrate, we upregulate this enzyme, and this may stimulate proliferation of memory cells that might explain this continuous increase in milk yield past the period of supplementation. So lastly, let me just point out that not only fat cows respond to choline. So we went back and mined our own data. So we had data from 230 cows from experiments that we fed choline starting prepartum. And we just asked the question, does it matter what body condition score the cow is when she enters the prepartum pen in terms of response to supplementing room-protected choline? So you can see here's the distribution of body condition score when cows enter the prepartum pen. Okay, for the control cows in one of the experiments, for the choline supplemented cows in the same experiment, and here's a second experiment here. So there's a distribution. We have cows that have low body score, I guess more what you would like to have in a well-managed farm that has excellent reproduction, so they don't come to the prepartum pen over-conditioned. And we had cows that were over-conditioned for sure entering the prepartum pen. So here's the distribution on histogram. So same thing, we have cows in a more ideal body score, and we have fat cows. So the question we ask, is there an interaction between body score and choline? Okay, so every dot here represents one or more cows. Okay, so this is, let's just look at energy-corrected milk. The blue dots represent the control cows, and the orange dots represent the choline supplemented cows. This is the body condition score prepartum. So I have here cows with low body score, moderate, and over-condition. You can see that the separation between the two lines, this is the line from the estimates of the statistical analysis of the linear model, the mixed model. And you can see that the orange is greater than blue, meaning that choline produced more than the controls. Okay, you can see here the statistical significance. And the second point, you can see that it didn't matter what the body condition score was. The response is present in skinny cows as well as in over-conditioned cows. So we didn't find any interaction between body score and response to room-protect choline. So what we think is happening is multiple things. It's not a simple just reduction in fatty liver. So when cows are fed choline, we don't see them mobilizing less body lipids. Okay, so they still have the same rate of lipolysis, to the best that we can tell from our own work and the work of others. So these lipids are mobilized, and eventually they have access to tissues, one of them being the liver. Imagine that this is the membrane of an hepatocyte, and there are transporters there that take these fatty acids into the cytoplasm of the liver cell. And then within the liver, those fatty acids bind to binding proteins that can take them to different directions. They can undergo oxidation in the mitochondria. If they are very long-chain fatty acids, they can undergo partial oxidation in different organelle peroxisomes that produces reactive oxygen species and chop down the carbon chain. So choline plays a role in the membrane of mitochondria, perhaps maintaining stability of the membrane and reducing the proton leakage that generates this reactive oxygen species. But those lipids can also accumulate in these lipid droplets here, which is the fatty liver that we see. But that is not just a simple accumulation of fat. These lipid droplets, as people study more and more, they're quite dynamic. They secrete molecules, they change in size, okay? They undergo degradation by proteolytic enzymes in the phagosome. And choline plays a role in stimulating, at least based on mRNA expression. So we don't have the definitive answer, but based on mRNA expression, stimulates synthesis of autophagosomes that can perhaps facilitate the degradation of these lipid droplets. But what we think it's really important is the assembly of this very low density lipoprotein to facilitate the export of triacylglycerol from the liver. There's data from different labs showing that cows that are supplemented with choline have upregulation of proteins that are important for the assembly and secretion of VLDL. Supplementing choline, as I showed you, increases the accumulation of glycogen. This has been shown in cows in vivo, and Heather has shown that in cell culture as well. Perhaps choline, through the action of phospholipids, phosphatidylcholine, improves the integrity of cells, including the gastrointestinal lining. Phosphatidylcholine has powerful anti-inflammatory properties, in addition to preventing or facilitating gap junctions and tight junctions to stay intact. And lastly, it may affect proliferation of mammary epithelial cells if this hypothesis is true. So these are some of the potential mechanisms that go above and beyond this lipotropic effect of choline. So just to conclude, I think hopefully I made the case that choline fits the criteria of a required nutrient for adult dairy cattle. Okay? So you reduce triacylglycerol infiltration. I didn't show you this data, but it improves feed efficiency in cows, about 60 grams more energy-correct milk per kilogram of dry matter. Obviously increases energy-correct milk. And it has some potential benefits to animal health based on the limited data in the literature. The response is influenced by the supply of methionine, probably because of the interplay between choline and methionine in this one-carbon metabolism, and in the synthesis de novo of phosphatidylcholine from S-adenosyl methionine donating methyl groups. So I think that makes a lot of sense, and the literature would support that based on what we found. The benefits are observed regardless of body scores, not just to be fed to fat cows. It should be fed to all transition cows. And the mechanisms are probably multiple. We simplify by saying that it reduces fatty liver. But there may be perhaps improved nutrient transport in the gastrointestinal tract, improved fat transport to the mammary gland because of the role on uptake of and transport of nutrients through synthesis of phospholipids. And there may be other non-classical mechanisms such as reduced inflammatory response and perhaps stimulate mitotic activity in the mammary gland. So thank you very much, and I think we'll take questions at the end, yeah?